Hello everyone, again welcome back to the latest lecture session. Again a quick recap of what we have been up to, right? What have we been up to? We have been looking at options for remediating contaminated groundwater, right? In that context I think we looked at mostly pump and treat and then containment too, and then I believe we looked at PRBs in great detail, and now we are going to look at the other major aspect here. Again we are dealing with contaminated groundwater and how do I remediate contaminated groundwater, right? So in this context, let's say I'm also going to discuss some other or non-technical aspects, but it's relevant obviously because the education is not just about gaining technical knowledge, but about understanding the relevant scenarios, let's say where you have the human element, yes, and try to apply your particular brains accordingly, right, if I may say so. Right, so in this context, I am going to talk about a particular scenario that I faced recently that's relevant in this particular context, let's say, right. So here I'm in Roorkee right now, right, and in this particular vicinity we have a river called Hinden, and I believe that more or less ends up joining river Yamuna if I'm not wrong, but you can check that. And Hinden river is one of the most polluted rivers in India now, let's say. And I think sugar mills, indiscriminate use of pesticides and what do we say, fertilizers, right, say, and you have different compounds, let's say, in these particular what do we say, contaminants that or different compounds in these particular products that you are applying to either the land or discharging into the water that act as toxic compounds or exhibit toxicity now, right. So again the key is that here we have what do we say, I think recently, let's say, a 2000 hospital bed was set up, and for primarily because exceedingly high, not exceedingly, pardon me, a remarkably high incidence rate of cancer in this particular vicinity or that particular area, let's say, right. So to address this, they set up this cancer hospital. Again obviously cancer is a symptom, and we people, at least to my knowledge, have not yet identified the source of this problem. But if you talk to people, people call a particular train as cancer train, as in where this particular train is mostly filled up of these poor people suffering from cancer, and the relevant people attending to the relevant particular cancer patient, taking the relevant patients to that particular hospital or from that, right. Again, as most people know, people suffering from cancer, I guess the treatment technologies are better right now, let's say, but still it plays havoc with the economy of that particular family and also the social integrity of that particular family, because the people attending to the relevant particular person are also going to be affected. Again, that's some aspect, because let's look at what I have here though. So here is the Hinden river, right. That's something we are talking about. You can search it, look it up on Google Map, right. But one particular aspect that came to my particular attention recently was this particular tributary. I think it's called Krishni or such. Again, you can look that up, right. This particular tributary, right. Again Hinden, I believe, ends up joining river Yamuna. Again, that's something that we see out here, right. And again, all this particular location, let's say, that I have out here is typically remarkably polluted, but it seems a lot of cases of cancer were higher, a remarkably high case of cancer were or are reported from this particular area. This is still, as of now in 2018 anyway, burning issue here, at least locally anyway, right. So again not much data that's available, at least in the public domain, but again it came to my attention because a few of my particular colleagues are from this particular area now. I mean, they are biotechnologists, right, and they are from this area, and that they looked at some preliminary studies, let's say, and they found that the concentrations of heavy metals, plasticizers, and some of the other pesticides and such were high, but they haven't looked at that in great detail yet, right. So let's say if you have such a case or a scenario, let's say, how are you going to try to remediate the particular site now? It's not one particular village that's affected, it's a cluster of villages, a wide swath of large swaths of land here that are contaminated. How would you go about it? Pump and treat? Nah, it's not going to work. PRB? Not really, right. Again, PRB, let's say if you have a landfill, let's say, that's unlined and you have leachate coming in, or you have relatively what do we say is the shape of the particular or size of the particular plume is relatively limited, yes, PRB is applicable and so on here. But what would you do when you have so such a wide area that's contaminated and especially leading to looks like carcinogenicity, let's say, right? Again, so here there are different aspects, as you can see in this particular stretch. We have a sugar mill, a dairy plant and a distillery drain, and they are discharging into this particular Krishni river, if I am not wrong. And then again more such mills, paper mills, sugar mills discharging into this drain, and widespread application of pesticides, let's say, in this particular area, and fertilizers too, right? Some of which can contain heavy metals, or maybe the heavy metals are inherent in that particular area with respect to, you have different strata. Sometimes you have arsenic, let's say, that is present in high concentration subsurface too, or relatively high concentrations. Again, different aspects, but this is what we have. So if you are the person, what would you do though, right? So again, for this obviously you need some background, but the issue is that typically how it works is that though most industries have their effluent treatment plants, all these kinds of industries, either the efficiency is relatively poor, let's say, sometimes, not sometimes, most of them have effluent treatment plants, at least at this stage of our progress, let's say, but the efficiency is poor, and typically the reasons are because, let's say, not a great lot of what do we say, incentive for maintaining the quality of water, let's say, for the relevant management of that particular company. And again, what's the reason? Because as of now we do not have, again, in our what do you say, I guess this is obviously the case in developing country as in India, we do not have such a wide monitoring network of what do we say, monitoring network that would give us an idea about the water quality, let's say, right? So some issue crops up, there is a public outcry, people go take the sample, let's say, or then CPCB or SPCB steps in or such. Typically SPCB, the state pollution control boards, depending upon the state, let's say, they are undermanned, as in if 100 people are required, typically they have 30 or 25 people. Obviously, to do the job, you need, the relevant manpower, and again, that's something that's missing. And again, let's say out here, let's say, we have different kinds of compulsions too, political or otherwise, let's say, right? So again, these are aspects that you need to keep in mind. This is the ground reality in general, let's say. And the issue here is that, let's say, even if the relevant management wants to get it done, or as in treat the water to the proper extent, typically, the relevant plant operator has limited understanding of the basic aspects, let's say. So if something goes awry or goes wrong, they try to apply, typically anyway, thumb rules and try to get things done, but they typically do not work. So again, it ends up being the case that the efficiency is not great. I mean, it's acceptable sometimes, but again, keep in mind that if you have so many industries that are coming in and and dumping their waste or treated waste or partially treated waste into the relevant waters, let's say, you are going to have a remarkably what we say, polluted stretch of the river. So in monsoon, you'll have the river flowing in and you have a lot of volume of water or discharge, let's say, and so typically dilution. But during the non-monsoon season, more or less, most of these rivers or streams, or most of these rivers, as we are calling it, are actually streams that just have the flowing waste, if I can call that. Obviously, where is that going to go to? It's going to seep into the groundwater, contaminate the soil and so on, and act as reservoirs, let's say, depending upon the type of compound, right? So that's something to keep in mind, let's say. So let's say, this is a huge area now, I believe this is in Uttar Pradesh, hundreds of square kilometers or such, let's say. So obviously, you need to look at a multi-pronged approach, at least in my opinion. I'm sure other people will have other opinions and so on and so forth. Right. So obviously, one aspect that we looked at recently or proposed was that, let's say, we are going to have to look at, let's say, upgrades for the relevant effluent treatment plants, such that even if the biological process that people use to treat these particular effluents do not work, they'll have other tertiary techniques that can be, let's say, relatively easily ramped up or such to be able to treat the water, right? So that's understanding the, or containing the source in a way, source in our context of the Indian context anyway, right? The other aspect is, obviously, you already have contaminated, or not obviously, most probably you have contaminated groundwater or soil now. How do I go about it? And obviously, people are mentioning that they have more or higher incidence rates of cancer. To my knowledge, not a lot of study is done to see if the cancer rates are relatively higher than what you would expect in a particular control population. But yes, here there is sufficient initial data to go by with that particular hypothesis. Again, so what do we need to do? Initially, obviously, we need to look at a monitoring study to understand what are the different compounds or contaminants. So upstream and downstream, let's say, as in I can't go to each industry, get the relevant data, analyze that. That's way too humongous a job, and we don't have the resources or the time or the manpower to be able to do that, right? And so we are going to try to identify the major sources, let's say upstream and downstream of the relevant locations maybe, right? And then with distance and sampling at various locations of our groundwater and soil. So once we do that, let's say we can understand what are the different contaminants present out there. Again, the issue here is that if I don't know what's causing the cancer, what are the typical contaminants, I need to look for many types of compounds. Heavy metals, let's say limited set of heavy metals. I can start with I think 23 or so, right? I know the major heavy metals of concern, so I can look for those. That's fine. But with respect to, let's say, pesticides or fertilizers or their byproducts, there are many such compounds that can lead to toxicity, let's say, right? How do I go about that? So obviously for that I need to have, let's say, LC-MS, GC-MS and so on, let's say. And typically they are around 60 lakhs or so, depending upon the type of vendor, 60 lakhs each of them, right? So again, the case here is that, to get something done, especially after you have relevant, contamination, the costs add up, especially in a country like India, let's say, where we are still developing the, the government is burdened with, not burdened if I say that it has many responsibilities, yes. So again, the funds available are limited, yes. So these are different aspects again, but I'm just giving an idea about the different costs here. Again, that's fine. So obviously we need to understand the, have a monitoring campaign initially, right? And from that I'll get the data regarding the type of contaminants, right? Yes, and then if I have relevant data about the toxicity, well and good. If not, I'll conduct the toxicity assessment. And then what can I do? I can conduct the risk assessment, let's say. This is something we discussed, right? So risk assessment, and then I can identify those locations within this stretch of area, let's say, and a generic, analysis, generic analysis obviously, because the data otherwise going into details for this stretch of or large areas, let's say, in my opinion, is relatively difficult to do so. So once you identify those areas that where the target population or is facing greater risk or unacceptable risk or remarkably high, highly unacceptable risk, let's say, you can target those locations for remediation, let's say, okay. So now let's say we have targeted those locations for remediation. How do we go about that, right? Because let's say if I'm a researcher, let's say if I want to go and get things done, that's not how it works. Why? I need some money. And how do I go about that? I go to different funding agencies and ask for money, let's say, right? And say that I'm going to propose this, so on and so forth. And based on that, I'm going to get things done if the funding is approved, let's say. But again, keep in mind that, depending upon the priorities of that particular funding agency, is it groundwater, surface water or science or such, each particular funding agency has their own priorities now. Let's say, I did approach or we did approach one particular funding agency, but they were receptive, yes. But obviously the experience that they were concerned with was, one of the aspects was adverse media, coverage, let's say. So typically in our context right now, we have the case where each and every aspect is drummed up, and thus, let's say, people are very much apprehensive about, getting things done, because if not getting things done, let's say, having adverse, data coming up, or data that's not, suitable to you, if I can call it that, or data that can have issues down the line or will require the relevant government or the people, let's say, to take some action, let's say, right? So people typically want to avoid such situations, right? So, and we have many such contaminated sites, or if not many, considerable number of contaminated sites in India. So again, it's an issue of priority. So how do they fund that? So, these are issues that come up whenever you go to, let's say, a funding agency or such too. So obviously these are aspects where the human element comes in, as in you have the media involved, and thus a local population, you have the NGOs involved, let's say. Obviously they play a very much worthwhile role, yes, but they have their own limited resources too, yes. So, and again, you have the relevant, what do we say, political pressure, right? So, that's obviously a fine balance between all these aspects now, right? Again, so those are aspects that I wanted to discuss, because obviously technical knowledge is relevant, but you need to be aware of the relevant aspects on the ground here, right? So, that's my experience very recently, and I wanted to just share that, okay? So, moving on though, how do you go about this though? Or how would you, get the system or make the system better, system as in, at least from a technical point of view, assuming you have most of what you want, let's say. So, obviously, we can have a multi-pronged approach. Obviously, we are going to look at, containing the discharge of the relevant contaminants. So, looking at improvements of the effluent treatment plants. And secondly, in this large scale, let's say if I have identified the relevant contaminated zones, remarkably contaminated zones, I look at PRB, maybe pump and treat, or some of the soil or sediment remediation techniques we are going to discuss later, right? But typically, let's say if it's been considerable time since it's been released, and if the usage, let's say, of fertilizers or pesticides, or let's say discharge of these contaminants is over a wide area, you are going to have issues with applications of most of these kinds of techniques. So, what do I need to look at? I need to look at bioremediation, let's say, bioremediation, or understand or discuss natural attenuation, let's say. Yes. So, first about natural attenuation, I will talk about bioremediation within the context of natural attenuation, right. So, I guess it is self-explanatory, it is natural attenuation. Attenuation as in, how do I understand that now? Let us say reducing the effects of some aspect or variable, let us say. So, here let us say nature, does its job typically. Given enough time, it is going to be able to degrade the relevant contaminants in that particular soil now, right. And again, there are different mechanisms and such involved. So, obviously, one of the mechanisms is that you have the microbes in the relevant nature, right, not in the relevant nature, in nature, and they are obviously going to degrade the relevant contaminants. Why is that? Because not obviously, they are going to have or they are going to try to degrade some of the contaminants. Why is that? Because similar to us, they are, as in the microbes, also need sources of energy for growth and cell synthesis, let us say, right. They want to survive, they want to grow, they want to thrive too, right. So, they keep looking for worthwhile sources of energy. And in the soil, let us say, if you have the relevant electron acceptor and donor, one of which can be your contaminant, the microbes will do their job and try to degrade that particular what do we say, contaminant. So, if I am not adding anything to this particular system in this particular location, and just trying to understand, let us say, or I have the relevant data, if not understand, and if I say that natural attenuation takes at such a rate that I do not need to do anything, within a reasonable amount of time, considerable swaths of the area will be what do we say, will witness a decrease in mass or concentration of the contaminant, right. What can I do? I can obviously model the relevant case, and then put forth data that natural attenuation is occurring at reasonably faster rates, right. And also that what is it now, that I have enough monitoring wells to be able to get this done, right. So, natural attenuation, obviously we just discussed in brief, let us say, one aspect with respect to what we say, degradation of the relevant contaminant by the microbes, but there are other mechanisms obviously. But in that context, I want to talk about bioremediation for a few more minutes, and then we will move on to the other aspects, I guess. So, let us see what we have here. So, what are the different attenuation mechanisms though, right. So, this is something we are going to discuss. So, one is non-destructive, obviously that means the next set of slides is going to be about the destructive mechanisms. So, in that the concentration is attenuated or concentration is decreased, let us say, right. So, keep in mind that the compound is not being degraded, right, but the concentration is decreasing. How can that happen? That means that you have dilution due to various reasons, right. So, let us say if I have 5 grams within 1 liter, it is 5 gram per liter, but then due to various reasons, let us say if this 5 grams is now present in 100 liters, obviously the concentration is less and the effects are less too. And but keep in mind that the compound, the total mass of the compound is still 5 grams in the total system, right. So, here we are going to briefly look at some of the non-destructive what do we say, mechanisms. Obviously, what are they? Think of what we have there. So, you have groundwater flowing through the particular what do we say, contaminated site, let us say, and if you what is going to happen, your groundwater is going to transport the contaminant, right. So, you are going to have dilution of the contaminant, right, because it comes in contact and you have advection, let's say, right. So that's one particular aspect. Or let's say as groundwater transports this contaminant, some of the contaminant can be adsorbed onto the particular soil, let's say, right. Again, so thus the concentration in the groundwater will decrease. Why is that? Because the contaminant is now adsorbed onto or can be adsorbed onto the relevant soil, let's say, right. And again you have dispersion because it's groundwater, not diffusion, but if I am going to use the term dispersion, let's say, right, to understand the tortuous path or effects of dilution due to the tortuous path, we will discuss this later in greater detail, or let us try to get this done. So if I have a particle here and this is the top view, let's say, and all these are my soil particles, let's say, soil, and this is my contaminant, let's say, right. As groundwater takes this contaminant through, there are different paths that this contaminant can take, right, or here, right. So you have a particular, so same contaminant starting at the same location, because of the tortuosity of this particular system, let's say, can be dispersed, right. And thus what do you see? It can end up at different locations. So thus again you see dilution or decrease in the concentration of the relevant contaminant, right. Again, what do we look at? This is my contaminant here. These are the different soil particles. This is the top view, obviously, right. And if the groundwater is taking this particular contaminant along with it, because of the tortuosity of that system, let's say, I am going to have dispersion, let's say, right. So let us look at some of the other mechanisms out here. So sorption or adsorption onto the soil, that is going to lead to decrease in concentration of the groundwater or concentration of the contaminant in the groundwater. I am going to have dispersion, that is something we looked at. Diffusion and dispersion, let us say, can happen. So I will say due to diffusion too, right. And then dilution, because you have advection, let us say, typically when I am talking about dilution, I am referring to the effects of advection, as in groundwater, what we say is taking the contaminant over what we say a larger area than it was initially present in. So the concentration is going to be lesser. Obviously volatilization, as in let us say you have, let us say, hydrocarbons or other such volatile compounds, let us say, right. So they are volatile, obviously. What does that mean? That they would want to stay in the gaseous phase rather than in the aqueous phase, right. So if I have gas and some air out here, and the relevant, not gas, pardon me, water, and the relevant contaminant which is volatile here, and I expose that to the gaseous phase, what is going to happen to the volatile compound? It does not want to stay here, it wants to stay in the gaseous phase, right. So it is going to change phase. By changing phase though, we see that the concentration in this particular water is decreasing. So obviously here we have different non-destructive mechanisms. Keep in mind that they are non-destructive, as in the mass of the contaminant has not changed, there is no degradation here, it is only that the concentration is decreasing. So that will have some effects, as in let us say if it is a wide area and I can't treat it right now, but I can see that after a few years or maybe months, let us say years maybe, right, the concentrations are low enough in the groundwater that they do not pose a major threat, let us say, right. So again natural attenuation takes place due to non-destructive mechanisms. So the other aspect obviously is going to be the destructive mechanism. In this case obviously I am going to witness both decrease in mass and thus concentration, right. So destructive mechanism, as in the compound is degraded, let us say, right. It is not that it just change phase from aqueous to the soil, aqueous to the air or such, right. So these are the aspects. So obviously how does that come about? You have biodegradation, which we are going to discuss in greater detail, right. And again biodegradation, what is the the driving force, pardon me, it is that the microbes want to thrive, right, and they need sources of energy, and your contaminant will act as either the electron acceptor or donor. As in when you have a redox process, let us say A plus B going to its oxidized and reduced forms, let us say products, you have energy released, right, and this particular energy is used by the particular microbes though. And how do the microbes facilitate this particular process though? They release enzymes that act as catalysts for this process to go through faster, right. So as in A and B can what do we say, just A and B in contact with each other can lead to, let us say, a redox process going through. But as you know, redox process, the issue is that time is an issue. You do not know if they are relatively fast or relatively slow, but typically though they are relatively slow, right. So, how do you want to, how do you facilitate this particular process to go through faster, or how do you facilitate this particular process? By adding a catalyst. So, that is what the role of the microbes are, that they are the release enzymes, let us say, that act as catalyst to make this process go through faster, and thus take the relevant energy for their growth and synthesis, let us say. So, biodegradation, and there are two forms typically. Aerobic, obviously, primarily let us say aerobic process. Aerobic means when the redox process in the presence of oxygen, let us say, right. And what is oxygen? Obviously, it is an electron acceptor, right, or an oxidizing agent. So, obviously, which particular compound will be degraded in aerobic conditions, let us say, right, a reduced compound or a electron donor. A redox process will go through only when you have both electron acceptors and electron donors, right. It is not that the half reaction will go through, you need to have both the electron acceptor, right, and also the electron donor. So, in that context, let us say in aerobic conditions, if you have electron acceptor, which kinds of compounds will be degraded? Those that can act as electron donors, or which are reduced compounds, let us say. And obviously, petroleum or hydrocarbons, right. Hydrocarbons, C and H, that obviously tells you that the carbon is in the more reduced form and thus can act as a electron donor, right. So, these are prime candidates. Again, we will come back to these aspects in greater detail, and then obviously we have the anaerobic conditions, right. So, here we see that I have the example for chlorinated solvents. Chlorinated solvents, as in they have Cl minus, let us say, or Cl, pardon me, right. And so, what does that typically indicate? That Cl is typically electronegative, or one of the more electronegative compounds out there. So, it is going to draw the relevant electrons towards itself, or pull the electrons towards itself. So, typically when you have chlorine, let us say, what does it mean? That the relevant compound are or is more oxidized, let us say, right. So, typically when you have oxidized compounds, you need anaerobic conditions for their degradation. We are going to see why, okay. And as we looked at, in aerobic conditions, typically reduced compounds are degraded, right. That is something that we just looked at, right. So, let us move on. And again, then abiotic degradation, as in you have two aspects out here, as in first, let us say a biotic degradation takes place, biotic process, and you have some byproducts, let us say typically hydrogen, or let us say ferrous iron is produced, let us say, right. And what do they act as? They then can act as reducing agents to degrade the contaminants. So, there is again a symbiosis between biotic and abiotic process, right. That is something we have here as an abiotic reductive dechlorination, right. So, that is something we are going to look at later. So, next. So, typically we are going to look at biodegradation for some time, or bioremediation for a few minutes, right. So, obviously as we discussed, you already have microbes out there, and you are going to have naturally occurring bioremediation, right. So, that is obviously within the context of natural attenuation. But what are the issues though, right? What are the issues here? And I think we discussed this result of metabolic activity of microbes, as in metabolic activity meaning the process that are involved in the production of energy required for growth, or sustaining life, right, metabolic activity. So, we have our own metabolic process, the microbes have their own metabolic process, and so on. So, obviously the driving force is metabolic activity, as we already discussed. So, again, what do they try to do? They need to get energy for growth and cell synthesis, right. The microbes want to grow. So, the key aspect that I was trying to hint at earlier was that kinetics is an issue, right. Depending upon the concentrations of the relevant acceptors, electron acceptors or donors, let us say, or because of the type of redox process, the kinetics can typically be low, not even for natural attenuation, let us say, right. Or it can also lead to incomplete degradation, as in let us say if complete degradation is represented by A going to Z, but it only ends up at A going to D, let us say, right. So, D might be more toxic or still toxic enough. So, incomplete degradation is also a relevant aspect, as in you will not have complete degradation. So, what do people typically look at? They look at biostimulation. In this context, what do they try to do? If your compound, let us say, is the electron donor, let us say, right. For the relevant process to go through, what do you need? You need a electron acceptor. And typical electron acceptor out there, what is that? Out there is oxygen, obviously, right? Or let us say similar to us, the relevant microbes also need the nutrients, let us say nutrients. The pH conditions should be suitable for their growth, and also let us say your temperature plays a considerable role here. And obviously they need a carbon source either from the donor or acceptor typically. In this case obviously from the donor, I guess, right? Carbon, nitrogen, phosphorus, the other nutrients and so on, let us say. So if any of these are missing, obviously the process will not go through. So in biostimulation, let us say you are going to add, or if there are any missing links, you are going to provide those missing links so that nature does its job. So obviously here what do we have? Addition of relevant oxidant or reductant and such to increase the bioavailability within the medium, right? And in that context we typically have bio-venting or bio-sparging. If it is in the vadose zone or such, bio-venting, sparging in the, what do we say, the groundwater and such. And I believe I have a graph here, okay, I have a particular schematic here. So typically what can we provide? We can provide air. When we say we are providing air, what does that mean? I am providing oxygen to the system, which is an electron acceptor, right? So if my particular contaminant is hydrocarbons, let us say petrol or diesel has leaked from my underground storage tanks or such, let us say, and is now an issue there. Hydrocarbons are relatively reduced forms of what we say carbon, let us say, right? And now you need an electron acceptor for the process to go through, but there is not enough dissolved oxygen in your particular groundwater, let us say. So what can you do? You can provide the oxygen so that the microbes present in that particular system can utilize the oxygen and this particular hydrocarbon, and then facilitate this redox process, thereby meeting both the needs, as in their need of energy and our need of degradation of the relevant contaminant. So here let us say what do you have? You have, I think, an example here, an underground storage tank here of petroleum hydrocarbons, and you have the relevant compound that is released. So 2 or 3 phases here. Vapor phase, obviously hydrocarbons are volatile, so that is what you see here. I think this is visible, this is the vapor phase, as you can see, and then adsorbed onto the soil, let us say in the groundwater. Before it reaches the groundwater, it is adsorbed onto the soil, and that is something right, they are relatively more hydrophobic, so considerable fraction would be adsorbed onto the soil, and if it reaches the groundwater table, it can also be have contamination of the relevant groundwater. So what do people do? So let us say we have a compressor, they have these particular wells put in, and you pump air or typically oxygen in this case through the particular system, let us say, right? And obviously here we see that there are 2 aspects here, rather than just biosparging, where you are putting oxygen through to facilitate remediation of this particular or natural or bioremediation of this contaminated zone. We are also pumping the gas out from this vadose zone or the vapor phase, let us say. So that is typically, let us say, soil vapor extraction. That is something we are going to discuss again later in the context of soils or sediments, as in here there are 2 aspects. One is you are providing oxygen to facilitate the growth of, or not the growth, pardon me, the degradation of this particular what do we say, the compound, which is hydrocarbon, and how does that go about? By the relevant microbes. And the other aspect that is going hand in hand in that, in this particular non-saturated zone, you have the relevant hydrocarbon, which is in the gaseous phase. So they are pumping that out, soil vapor extraction, that is what you see here. So vacuum conditions are created here, and you pump the particular compound out, and then vapor treatment, and then the vapor is discharged, I guess, right. So it goes hand in hand, let us say, as we mentioned earlier. So bioremediation, and in that context biostimulation, as in we are providing one of the missing links, right. So I am going to move on. So bio-augmentation, so at least in the Indian context, it I think rarely has been looked at, but I think abroad or such there has been considerable, what do we say, application of this bio-augmentation, right. And what is this about? Layman's terms, let us say, right. So let us say depending upon where I have grown or such, I am acclimatized to a particular kind of environment, let us say. Either maybe I can tolerate heat pretty well compared to other person, but the other person can tolerate colder weather pretty well, and so on. So if you take me and put me in a relatively cold place initially or such, or even generally, let us say, I am going to struggle, let us say, right. So here I am talking about acclimatization. So here let us say you have relevant compounds, toxic compounds, and toxic to us, they are also at a high enough concentration toxic to the microbes too, right. But they might not be toxic to all the microbes, there might be certain kinds of microbes that would want these particular contaminants or use these contaminants for their so as their sources of energy, let us say, right. So what would you do though, right. So here what can you do? You can try to isolate the microbes from those locations where the contaminant concentration is very high, let us say. I think we looked at one example in Ghaziabad, right, where we had chromium, what do we say, contamination. So what did people do? They looked at or isolated the relevant microbe strains from a particular site somewhere else, let us say, where the chromium concentration was very high, right, that most other microbes were not thriving, but a particular type of microbes were thriving. So they isolated those microbes, they cultured them, grew them, if I can use the layman's terms, let us say, and then introduced those cultures in this particular chromium contaminated groundwater in Ghaziabad. So that's more or less, if I can say, about bio-augmentation, as in I am going to introduce non-native species which have a preference, let us say, for degrading those particular contaminants into my particular contaminated site. That is one way. And the second way is to also engineer, let us say, or change the DNA of this particular microbes, let us say, such that these engineered microbes can facilitate the removal or degradation, pardon me, of the relevant contaminants. So they are the engineered microbes, and you can engineer and put those microbes in, but again it is a costly affair, and from what I have known, let us say, the chances of success are relatively less. And again the reason is that obviously there is a particular reason why the microbes that are in that site are thriving. Why is that? Because it is the principle of, if I may say, survival of the fittest, right. So they are the fittest for those conditions now. So if you put some other particular microbes in, it does not necessarily mean they will thrive there again, right. So again you might need to change the conditions or such, or these microbes, let us say, can feed on these other non-native species or the engineered microbes, let us say, or these engineered microbes which do well in the lab conditions might not end up doing well or might not acclimatize themselves to these particular site conditions. Again, bio-augmentation in a nutshell, if I can say, right. So analogies to engineered systems though, right. Again, as in what are we doing in this bio-augmentation? We are bringing in microbes, let us say, if I can use the layman's terms, and introducing them here. So engineered systems, as in you have your effluent treatment plants, So where do you see the relevant analogy though? As in you have your activated sludge process, ASP. So what is that about? You have your wastewater coming in, and you add oxygen here, which is your electron acceptor, right, and this is your aeration tank, that is what we typically call, and then your secondary settling tank, and the sludge is again recycled, right. It is activated sludge, as in this activated sludge is recycled again, and then the supernatant, which is the water, is the cleared water or the treated water. So what is happening here? This sludge contains the relevant microbes, let us say, and these microbes are now again recycled into the system, such that you have a relatively high concentration of the microbes, or those microbes which can treat your or degrade your waste, and oxygen is your electron acceptor. So you can think of these as analogies, let us say, right. So in engineered systems, as in our ASP or SBR, we are already looking at these cases as in promoting the growth of a certain kind of microbes, let us say, that would promote degradation. So you can try to understand that in terms of bio-augmentation, I guess, right. So moving on, let us say, and we are going to move on to looking at the evaluation, advantages and disadvantages, let us say. Obvious advantage is that, let us say, the costs are relatively less, right, but obvious disadvantage is that the kinetics are typically slow, especially for only natural attenuation. That is why you either try to stimulate it or introduce the non-native species. But there are some other advantages and disadvantages. We are going to discuss that in the next session, because looks like I am out of time. And then we are going to look at the mechanisms, let us say, some of the mechanisms, and then move on to the relevant models with respect to how do I predict what is going to happen to the system, right. For example, I can't just say, okay, this looks like a good candidate for natural attenuation, and I am not going to do anything and just wash my hands, right. Obviously, I need to be able to predict what is happening and then show that, okay, natural attenuation is occurring, or if that is not good enough or the rates are not good enough or such, then I am going to look at providing the stimulus, let us say, or stimuli to fasten the process, right. So I guess with that I am going to end today's session, and thank you.